Hello everyone! Today our video's theme is to introduce the earliest gene sequencing technique, first-generation sequencing technology, also known as Sanger sequencing. While this method has gradually taken a backseat with the evolution of sequencing technologies, it remains the gold standard due to its exceptional accuracy. In this video, we will delve into the origins of this technology and its associated operational processes. So let's get started with today's video. First-generation sequencing, also known as Sanger sequencing, is a DNA sequencing method that involves electrophoresis. It is based on the random incorporation of chain-terminating dideoxynucleotides by DNA polymerase during the in vitro DNA replication. Invented by British biologist Frederick Sanger in 1977, it is named after him as Sanger sequencing. This method has provided a powerful tool for understanding the importance of DNA sequencing and addressing genetic questions. Sanger sequencing laid the foundation for modern genetics research. Sanger DNA sequencing is widely used for the following research purposes: verification of next-generation sequencing NGS results, confirmation of plasmid sequences, inserts, and mutations, HLA typing, genotype analysis of microsatellite markers, identifying genetic variations responsible for single-gene disorders. Using the chain termination method, fluorescently labeled dideoxynucleotides, ddNTP, are inserted into the newly synthesized DNA chain by DNA polymerase during DNA replication, causing chain termination. These terminated bases are differentiated by different fluorescent colors. The synthesized DNA fragments are separated by size on a polyacrylamide gel, and a fluorescent laser scanner is used to detect the position of each termination and its corresponding color. By collecting data from the fluorescent signals, the DNA sequence can be determined, as each termination position corresponds to a specific nucleotide. Next, let's delve into the detailed steps of how first-generation sequencing is performed in DNA sequencing. 1. Sample preparation. Firstly, the DNA sample to be sequenced needs to be obtained. This sample can come from various sources such as blood, saliva, or tissue biopsies. 2. Fragmentation. The DNA sample is broken down into smaller fragments, typically ranging in length from about 100 to 1,000 base pairs. Fragmentation is crucial to make the sequencing process more manageable. 3. Primer annealing. Primers are short synthetic DNA sequences specifically designed to complement the target DNA region intended for sequencing. 4. Extension. After primer annealing, the next step is primer extension, during which nucleotides are added to the primer to synthesize a complementary DNA strand. These are the initial steps in the process of first-generation DNA sequencing, setting the stage for further sequencing and data collection. 5. Termination. During the synthesis phase, a class of nucleotides called termination ddNTPs is introduced into the reaction mixture to terminate synthesis. 6. Gel electrophoresis and imaging. The reaction products are separated by size using denaturing polyacrylamide urea gel electrophoresis. After gel electrophoresis, the DNA fragments are visualized using a process called autoradiography. The final sequence is assembled. These steps complete the process of first-generation DNA sequencing, where termination of synthesis and gel electrophoresis are critical for determining the sequence of the DNA. Currently, there are several products and platforms based on Sanger sequencing technology available in the mainstream market. Here are some of the key Sanger sequencing products and platforms. ABI 3500 or 3500XL Genetic Analyzer. The ABI 3500 or 3500XL Genetic Analyzer, introduced by the biotechnology company Applied Biosystems, is a widely used Sanger sequencing instrument. It offers high-quality sequence data and is capable of achieving longer read lengths. Beckman Coulter CEQ 8000. The Beckman Coulter CEQ 8000 is a Sanger sequencing instrument based on capillary electrophoresis. It features automated operation and rapid performance. Since its development in the 1970s, Sanger sequencing has been a cornerstone for molecular biology and genomics. This reliable and accurate method has facilitated numerous discoveries in the fields of genetics and molecular biology, playing a crucial role in projects such as the Human Genome Project. While next-generation sequencing technologies have emerged and replaced Sanger sequencing in many applications, the latter remains relevant for targeted sequencing, validation of specific mutations, and other specialized cases requiring high accuracy and longer read lengths. Sanger sequencing will continue to be an indispensable tool in the molecular biologist's toolkit.